Hi there guys, welcome to lesson 7.8. In this lesson we'll be talking about nematophagous fungi, and these are fungi that consume or eat nematodes. So we've turned the tables now from nematodes that consume fungi to fungi that consume nematodes. In this lesson we'll cover various mechanisms that fungi have in order to either defend themselves from being consumed by nematodes or the mechanisms they possess in order to actually consume nematodes. The image on the left shows fungal hyphae coming out of a nematode that the fungus has consumed, and these are the kind of horror stories you'll be hearing about in this lesson. Okay, so let's jump into lesson 7.8. So fungi don't just allow nematodes to eat them. They have their own fungal defenses as a resistance mechanism versus nematode predation. And that can happen via toxic chemical defense, by the production of proteins, peptides, pigments, and various secondary metabolites. And examples of this are biotin-binding proteins, lectins, protease inhibitors, pore-forming proteins, and ribotoxins. These toxins bind specific target molecules on the nematode, so they can act as deterrents or inhibit viability of the nematode or its growth and development. An example of this is Coprinopsis cinerea, which is featured here on the right, and it produces mycelial surface toxins. The hyphae, the fungal cells themselves, can be covered with toxins, and these are cytoplasmic lipases, so they bind to lipids or fats on the nematode and they interact with those lipids, and because they are lipases they actually degrade the lipid, so that means that they can kill the nematode upon contact because they degrade the fatty component in the nematode cuticle. Another example of a toxin is that of oxalic acid in Aspergillus niger, and it's toxic to Aphelingus aveni. Arbuscular mycorrhizal fungi also produce isoflavonoids, and these increase plant resistance to root-feeding nematodes. So these are just examples of the various ways that fungi actually defend themselves against nematodes or are toxic to nematodes. However, some fungi actually take it a little bit further. They actually become nematophagous. So that means that they consume nematodes. So instead of just defending themselves or being prey to nematodes, they actually go out of their way and create various mechanisms in which they can actually ensnare and digest nematodes. And some of these nematodes are nematode trapping fungi, while others are endoparasites of nematodes. Over 300 species now are reported to have been identified, but there are up to 8,000 of these fungi waiting for identification. These fungi exist across all the major taxonomic groups and on all continents and in all soil environments. And because many of them rely on organic matter as their main food source, they do prefer soils that do contain organic matter. So basically they can be classed as nematode predators, but really the reason why they develop this feeding behavior is to actually access nitrogen in nitrogen-limiting soils. So majority of them are opportunistic, with the exception of endoparasitic fungi that actually do require a nematode for the active part of their life cycle within the nematode. Some of them are ovicidal, so that means that they actually consume nematode eggs or nematode cysts as well. And the endoparasites are fungi that actually use their spores as a mechanism to get inside the nematode and then develop and grow within the nematode. And then there are nematode trapping fungi, which produce special attack devices, and that includes traps and even weapons. And then some are toxin producing. And these toxins are nematocidal or nematotoxic, and they paralyze the nematode, and the fungi then are able to consume the poor creature. So the diagram on the right shows these different nematode consuming capabilities of fungi. And that includes the nematode trapping fungi. They actually create these elaborate structures to actually entrap nematodes. The endoparasites that have their life cycle within the nematode and then grow out of the nematode. There are the egg parasitic fungi that consume eggs, and the toxin producing fungi that stunt or paralyze the nematode and then consume it. And there are also nematodes that produce all kinds of attack devices that are quite harmful to the nematode to kill the nematode before they consume the creature. So let's now look at some of the ways in which fungi consume nematodes. And we'll start with fungi consuming nematode eggs first of all. So this research has looked at how the Mortierella globalpina fungal hyphae consume eggs of Meloidogyne chitwoodi. So basically these are scanning electron micrographs that you can see here on the right. And in A and B, the hyphae of Mortierella are penetrating the egg. And you can see the hyphae actually colonizing the egg here. And then in images C and D, you can see how the cuticle or the shell of the egg has basically been devoured, and now the hyphae are moving on to consuming the contents of the egg. So this particular research was looking at how the root knot nematode, the Meloidogyne species of many different crop families, including Solanaceae, so like potatoes and tomatoes, Brassicaceae, so like the cabbages and broccoli and things like that, legumes, Cucurbitaceae, which is the pumpkin and cucumber family, as well as Poaceae, which are grasses, but many other families of plants in cropped situations are affected by the root knot nematode. So obviously research is focused on how to get rid of this nematode, and the biological method of control can be a fungal method. But as always, I will go on to say that when the ecosystem in the soil is in balance and everything works well and all the organisms are in the right proportions, the root knot nematode wouldn't be such an issue. And of course, if there are lots of fungi in the soil doing their job and there's a lot of organic matter, then the soil ecosystem is more likely to support a healthy plant growth, and there will be more natural resistance to any nematode or other organism to consume that plant. Some nematodes can be classified as endoparasites. So these are nematodes that actually have a majority of their life cycle within the nematode, so they grow as long as they are inside a nematode, and these are the obligate endoparasites. So that means that without a nematode, these fungi cannot complete their life cycle, and they will reproduce spores to actually release to the outside, but can only grow by consuming nematodes. Some of them are knee-obligate parasites, so these can, at least to some extent, live outside of the nematode, consuming other organic matter. And the way that these guys actually get inside the nematode is through producing spores, which are called conidia. They are asexual spores, and they basically have adhering structures on the spores that cause the spore to attach to or stick to the nematode. Some of these spores will germinate and the hyphae will grow into the nematode, while other spores actually inject their contents once contact with the nematode is made. The other way in which conidia or fungal spores end up in a nematode is through the ingestion of spores. So the nematode actually has to consume the spore. Then that spore will germinate inside the nematode, and then the hyphae will ingest the contents of the nematode and will actually consume the entire nematode. The genus Harpasporium has conidia that are shaped in a way so that they actually lodge in the buccal cavity or in the mouth or the esophagus of the nematode, whereas Drachmeria conispora have conidia that actually adhere to the chemosensory organs of the nematode. So basically the nematode cannot sense anything because the actual conidia, the spores, are actually attached to the structures or near the structures by which the nematode needs to function through the reception of chemical or other signals from its environment. So these are some of the ways in which the endoparasites actually make initial contact with their host nematode. So this is the infection cycle of Drachmeria coniospora. And the way it begins is that the conidiophores, these are the structures on which the conidia or the asexual spores are formed on, which have been developing on the inside of an infected nematode, come out through the nematode surface and form these spores. When the conidia are mature, they develop these spherical adhesive knobs, and the conidia remain dormant until they attach through this adhesive knob to a new prey. So as the nematode is moving through its environment, it comes upon the conidia, and these conidia with the adhesive knobs attach to the nematode near the chemosensory organs on the head. So this is the region where the nematode senses its environment. So it becomes really compromised by having these conidia sticking out of its head. And then there's a chemical interaction that goes on between the nematode and the conidia, and this allows a penetration of the cuticle through enzymes and the force of the aprosoria. So aprosoria are specialized structures that develop once the conidia germinate, and these aprosoria basically penetrate the cuticle, and the enzymes allow the dissolution or the degradation of the cuticle as well. So that then together allows the entry of the fungal aprosorium, and therefore the development and vigorous growth of the hyphae of the fungus in the pseudocols, that is just the body cavity of the nematode, as the hyphae feed on the nematode internally. And that, of course, then leads to death of the nematode. And once that happens, of course, the food source is running out. So now the new conidiophore has to form. And this is an image here showing how this new conidiophore is pressing on the cuticle internally, but does not penetrate the cuticle. So the contents of the nematode is still feeding the development of the new conidiophores. And then these come out again out of the nematode. So once again, we have the conidiophore with the conidium forming on the outside of the nematode, ready to mature and infect a new host. And once conidiophores are developed, they actually yield up to 10,000 conidia, so that the conidia can infect new nematodes. And so the infection cycle begins again. Other fungi create special structures in order to damage nematodes and enter nematodes to ingest their contents. An example of this is Coprinus or Coprinus comatus, and these fungi form what scientists have termed spiny balls. And this was discovered in the first instance in 2004 by visualizing these little balls with lots of spines on them. And so these structures have been shown to create many small cuts on the cuticle of the nematode, and these cuts then accumulate and therefore cause severe cuticle damage through the leakage of the inner nematode contents, as the nematode has a high turgor pressure, and that's how it moves. The cuticle is pressurized, so it's like a pressurized exoskeleton. So whenever there is a puncture in that cuticle, the pressure within it will cause a rapid leakage of its contents. In addition, Coprinus comatus also has been found to produce 7 nematode immobilizing toxins. So these two mechanisms, the damage to the cuticle as well as the toxins, work hand in hand to damage and immobilize the nematode, and then the fungal hyphae penetrate into the nematode and consume the poor creature. And the way that the hyphae actually penetrate the nematode once the spiny structures have caused the damage is through penetration pegs. And here we have Panagrellus redivivus nematode that is being colonized by hyphae of Coprinus through these penetration pegs, and you can see them going in here. And this will be probably associated with some enzymes secreted by these pegs to penetrate the cuticle of the nematode. And this image shows the holes that are created within the nematode through these hyphae that penetrate its cuticle. And then the nematode is basically digested and consumed within days once the hyphae get inside. Another species of fungi that create nematode damaging structures is Stopharia, and this species creates acanthocytes, and these are structures that have little spines on them as well called acanthi, and they pierce and immobilize the nematode. This leads once again to leakage of the contents of the nematode and the decomposition or digestion of the nematode within 12 to 24 hours after immobilization and death. So it's a very rapid process. And within 36 hours, hyphae grow out of the digested nematode, and within 48 hours, there is a complete digestion of the creature, and this leads to further production of acanthocytes and hyphal growth. So we have come to the end of lesson 7.8, and in our next lesson we'll continue the horror story of how fungi consume nematodes. We'll look at the ingenuity, the elaborate mechanisms of the nematode trapping fungi that actually create specific structures to actually ensnare or entrap the poor unsuspecting nematodes. Okay, so you'll be hearing from me again in lesson 7.9. Bye for now.